Welcome to the sixth in the PSC Partners 2020 Annual Conference Webinar Series, Expert Updates in Liver Transplant, Clinical Trials, and Future Research Trends. Thank you so very much for joining us. My name is Mary Vyas. I'm the President of PSC Partners Seeking a Cure Canada, a Canadian registered charity and affiliate of the US-based PSC Partners Seeking a Cure. As a parent to a young adult with PSC, the topics for today's webinar are ones that I try to keep up with because they are sources of hope for everybody living with PSC. Transplant, because it can be life-saving. Clinical trials, because they are necessary evidence for approved treatments. And future research trends, because it is integral to the PSC Partners mission to fund research towards better treatments and a cure. It is also a tradition at each annual patient conference that attendees have an opportunity to hear from a member of the Scientific Medical Advisory Board on what is happening in the PSC research world. And so it is with great pleasure that we bring you this virtual offering of this very important conference session. Okay, let's get started with a poll to see who is here. Can we launch the first poll, please? Okay, the question is, I am a, and the answer responses are a PSC patient, PSC post-transplant patient, parent of a PSC patient, spouse or other caregiver of a PSC patient, or other. So it looks like most here are PSC patients, almost 40%, with the next parent of a PSC patient at about 30%. And then we have some post-transplant patients, some spouses, and some others. Thank you so much for participating. Thank you again for joining us. I'd like to pass the Zoom microphone to CEO and founder of PSC Partners Seeking a Cure, Ricky Safer.
Thank you, Mary. I too would like to welcome everyone to the sixth webinar in our series. All our past webinars have been recorded and are accessible on our website. After tonight, we will have one more webinar left, and we will be announcing it soon. In addition, we have scheduled a Zoom room event on October 6th for all interested caregivers. It's hard to believe that summertime is behind us. If you're like me, every day during the pandemic is feeling like Groundhog Day and time has lost its meaning. I miss all my daily social interactions as communicating on Zoom just isn't the same. I recently came across a meme which sums it all up. Me, this show is boring. Colleague, again, this is a Zoom conference. Tonight's Zoom conference, however, promises to be an enlightening and stimulating event. Our four expert speakers today will present updates on three topics of great interest to all of us: liver transplantation, clinical trials, and future trends in PSC research. Remember to send in your questions on the Q&A icon. We greatly appreciate the participation of Drs. Pomfret, Miller, Hirschfield, and Forman, and we thank you for sharing your wisdom with us. I'd also like to thank our conference co-hosts, University of Colorado Health and Children's Hospital Colorado, as well as our other corporate sponsors, Gilead, Mirum, Intercept, Albareo, and Shire, for their support of our virtual conference series. Just a few reminders about upcoming programs where we really rely on the participation of our PSC patients and caregivers. Most importantly, if you haven't already, please save the date October 23rd on your calendar to join us for Making Our Voices Heard, Virtual Patient-Focused Drug Development Forum. This is a one-time meeting approved by the FDA and led by PSC Partners. It's our big opportunity to shout out PSC patient voice, our chance to tell our patient stories, explain our symptoms and their impact on our lives, and also to identify priorities for future treatment. We need as many patients and caregivers to join us as possible to show the FDA the strength and dedication of our community. We'll be posting sign-up information on our website later this week. There's still time to fill out our PFDD survey, which will provide valuable data for researchers, drug developers, and the FDA. It will take just 30 minutes or less of your time, so please go to our website to fill out the survey sometime within this week and preferably go through our registry. We especially need more male PSCers, as always, and also pediatric patients and caregivers to complete the survey. We have recently launched our updated PSC Partners patient registry, which is filled with new resources and features. We urge you to complete or update the registry clinical survey and explore the new interactive features of our registry. If all of us as patients and caregivers do our part as a community, we can display the true power of our patient voice. So please join us. Here's a polling question for our participants tonight. Please launch the question. Okay, we just want to know, have you ever participated in a clinical trial? And caregivers, you can answer for your PSCer, either yes or no. All right, looks like 14% have enrolled in a clinical trial, which is pretty good because we haven't had too many clinical trials for PSC so far. So that's a good percentage for tonight. Now I'd like to introduce our moderator, Dr. Lisa Forman, Professor of Medicine, Gastroenterology Hepatology Program Director at University of Colorado Anschutz Medical Campus. I'll turn it over to you now.
So welcome everyone. Again, I'm Lisa Forman, one of the hepatologists at the University of Colorado, and I'm so excited to be moderating this webinar tonight. We were so disappointed that COVID disrupted our plans to host the 2020 annual PSC conference, but I'm very happy at least to have the opportunity to moderate and participate in these webinars. We have three amazing panelists today, Dr. Pomfret, Dr. Miller, and Dr. Hirschfield, who are all experts in their fields. And just again to recap, each talk will be 20 minutes, followed by a Q&A session at the end of all three talks. Please again limit your questions to general questions and not any specific questions that apply specifically to you or your loved one. And thanks in advance for PSC members who have submitted questions in advance. So with that, I'd like to introduce Dr. Elizabeth Pomfret. She is Professor of Surgery, the Egal Kalm Chair of Transplantation, and the Chief of Transplant Surgery at the University of Colorado. She's an international leader within the fields of liver transplantation, has advanced the field tremendously, especially within the area of live donor liver transplant. I have the honor of working with her every day, and I'm excited for all of you to hear her speak. Dr. Pomfret.
Thank you, Lisa. Thank you for the invitation, and thank you to the PSC organization for inviting me to talk today. So I am going to talk to you a little bit about liver transplantation, specifically about the workup, the evaluation for the donor and recipient. And then we're going to talk specifically about living donation and the outcomes associated with it. Next slide, please. So the natural history of primary sclerosing cholangitis, as most of you are well aware, is a progressive in nature and usually slowly progressive in nature, but it can have a highly variable course. Usually it progresses over the course of decades. There's no spontaneous cure or resolution of the disease. Some patients who have primary sclerosing cholangitis also have ulcerative colitis, and the primary sclerosing cholangitis is not affected or altered, the course is not improved by having your colon removed, so a colectomy. So that's sometimes something that people wonder about. And patients who are asymptomatic, in other words, have no symptoms at all, but just have the diagnosis, appear to have a better survival than those patients who do have symptoms. Next slide. So when is it time to consider a liver transplant? Next slide. It's difficult to say absolutes here, but to give you a general idea, about half of our patients require a transplant within 10 years or 10 years after diagnosis. So that means half of the patients don't require a transplant. Next slide. And as we discussed, since the course of the disease is very slow and insidious, often people don't realize how sick they've become And you often hear patients with PSC saying, well, you know, I'm really not that sick. I don't think I need a transplant yet. But we know you're becoming more and more sick when you begin to become jaundice, which is that yellow coloration in your eyes and your skin. You start having issues with itching when you require repeat procedures, ERCPs, to deal with maybe episodes of infection in the bile ducts called cholangitis. And that's when really you've moved from just you have PSC and you're fairly well compensated to now there are clearly symptoms associated with the PSC that warrant the need for discussing transplantation. Often we describe it as kind of sitting on a precipice or sitting on a cliff where the person is very, very stable, and then there may be a particular event that occurs, an infection or something that all of a sudden, you know, the person just really sort of dips right off that cliff. And that's when we are really in a need for urgent transplantation. And so at that point, we see that the patients are much more jaundiced. They have issues with their ability to clot. They have much more problems with itching, often with repeat infections, with weight loss, with admissions to the hospital, and generalized fatigue. Next slide. So the indications when we start to think about putting a patient through a transplant evaluation is when the patient has had repeat bouts of cholangitis or infection in the bile ducts, repeat stenting procedures or having percutaneous drains put in where the radiologist puts a drain in from the outside into the biliary tree. Having an associated cancer in the bile ducts called a cholangiocarcinoma, developing cirrhosis and the complications from cirrhosis, having a MELD score, and we'll talk more about what that is, of greater than 15. And there are certainly other indications. These are just some of the general indications where we would certainly put a patient through a transplant evaluation if they've had these different events occur. Next slide, please. So how will it happen? Well, when we actually do the transplant, and we'll talk in more detail about this, we kind of have two options. We either have a deceased donor, which is typically a full-size liver from a person who has died in such a way that their organs are still healthy and able to be used, but either they're brain dead or their care has been withdrawn and their heart stopped beating in a short enough period of time that we can use the organs. So that's deceased donation. And then living donation is when we take a portion of a healthy person's liver, and typically for an adult to adult transplant, we're talking about taking about 60% of the healthy person's liver, taking the recipient's entire liver out and replacing it with that partial liver. For a child, we would need less liver than 60%. For a baby, a much smaller piece, about 20%. Next slide. So the recipient evaluation for the patient who has primary sclerosing cholangitis and is undergoing evaluation for transplant has many parts to it and involves many different disciplines of medicine. There are the typical evaluation things that are done of getting a history and physical and noting all of the patient's medications and whatnot. We also do an assessment of the recipient's heart and lungs to make sure that there's no issues there that would preclude transplantation or that need some sort of treatment prior to transplantation to optimize those functions. Depending on what the recipient's age is, there are certain tests that we do, like colonoscopy, mammography, pap smear, that would be done prior to transplant to make sure that there are no other comorbid diseases. We have a whole slew of labs and imaging studies that get done. And then the recipients meet with financial counselors, with a social worker, with a psychologist or psychiatrist. There's a very robust patient education portion to this. And if the recipient has issues with drug and alcohol use, that would be part of the assessment as well. The surgeon assesses the patients for suitability as a surgical candidate, and there's a nutritional assessment as well. Next slide, please. So as I said, there are many different disciplines that interface in evaluating a potential candidate, and this is just an illustration of all of those different disciplines. And typically the evaluation will take place over a 2-day period of time. Next slide, please. So what is the MELD score? Well, the MELD score was originally designed for estimating the likelihood of surviving short term in a patient with cirrhosis who had undergone a TIPS, or a procedure where the portal blood system bypasses through the liver into the vena cava. And that score was initially developed by the Mayo Clinic, and then we came to realize that it had utility in assessing patient survival waiting for transplant. So in 2002, we began to use the MELD score as an objective way to be able to stratify patients who are waiting for transplant from the least sick to the most sick. And it's objective because we use laboratory values instead of subjective things like, well, they have a lot of ascites or not so much ascites. So we use the bilirubin, one of the clotting labs, which is called the INR, creatinine, which is kidney function, and the sodium. Next slide. Those values get put into this complicated equation, and then we derive a MELD score from that. And the MELD score goes from a minimum score of 6 to a maximum score of 40, 40 being the most sick, 6 being the least sick. And there are many different MELD calculators online that you can use to determine what your MELD score is. Next slide. The good news is that the outcomes after liver transplantation, all comers, is excellent. So the 1-year patient survival after a liver transplant is over 90%. The 2-year survival is about 84%. And you can see even when we get to 10 and 20 years out from transplant, there's still a good survival. And I would say pay less attention to the 10 and 20-year survival, because what we have now in terms of improvements in immunosuppression and in treating hepatitis C really make a difference in terms of what we're going to see for long-term outcome moving forward. Next slide. So as I said, after liver transplant, the estimated 1-year survival overall is about 90%. And for patients who have primary sclerosing cholangitis, it is possible for that disease to come and recur in the new liver. About 20 to 30% of the time, the patient can have recurrence of their PSC. And it might even be a little bit less than that, because I think if we look at the historic literature, often what we were attributing to recurrent PSC might have just been chronic rejection. And as I said, we have much better anti-rejection medicines now than we did, even 5 and 10 years ago. Even if a patient does have recurrent PSC in their new liver, most of those patients do well. And we are able to treat it, but sometimes patients require another transplant. Next slide. So the issue really is that there are more patients waiting for a transplant of any organ, but here we're speaking, tonight specifically about liver transplantation, but this is just the overall waiting list for any organ. And you can see that the number of people waiting far outstrips the number of organs available. So there is a definite gap that needs to be closed. Next slide. And this is the most recent data. We collect this data nationally, and what you can see in these slides is the slide on the left shows you that the blue bars are deceased donor liver transplants and the orange bars are living donor liver transplants. So as you can see, the vast majority of liver transplants that are done in the United States are deceased donor liver transplants. And the graph to the right shows you that, you know, what we worry about the most is the patients waiting on the waiting list, because the reality is that many patients will become too sick or die prior to actually getting a liver transplant. And you can see that nationally, that can be upwards of 2,500 to 3,000 patients. Next slide. So that really brings us to why we should talk about living donor liver transplantation, because living donor liver transplantation allows us to bypass the national liver allocation list. Now, because it's a partial liver, we cannot really use a partial liver when somebody is at death's door or has an extremely high MELD score and has other organ failure, like for instance, kidney failure. So we want to isolate those patients who we know who need a transplant, but their MELD score really isn't high enough to put them in a competitive range for a deceased donor organ. And so we don't want to transplant somebody too soon. So typically we use the MELD score of about 10 to 12 as our cutoff. For below that, you're probably at more risk having a transplant than waiting. And if you have a good organ or a potential living donor, then really a MELD score as low as 10 to 12, you could consider transplantation if that patient has had complications that aren't necessarily reflected in their MELD score. So recurrent bouts of cholangitis, recurrent hospitalizations, malnutrition, intractable itching. So those are some of the things that we take into consideration when we're talking about living donor liver transplantation. Next slide. So who can be considered as a living donor? Well, the donor has to be between the ages of 18 and 55. They have to be very healthy. They can't have any major medical problems, major psychiatric problems. They have to be thin. Typically, we say with a body mass index less than 30, and they have to be a compatible blood type, but not an identical blood type. So what I mean by that is blood group O can donate to anyone. Okay, that's the universal donor. So let's say the recipient is blood group A. If they're waiting on the deceased donor national list, they can only get a liver from someone, a donor who is blood group A. So A goes to A, O goes to O. And the reason why is because if we used O's for all of the other blood types, we'd never transplant patients who have blood group O and are the recipient. But with living donation, we don't have to adhere to those rules. So if you are blood group A as the recipient, you could receive a liver from a living donor who's either blood group O or blood group A. Next slide, please. Now, as you saw in that one slide, despite the fact that there's very good outcomes with living donor liver transplantation, it's only done about it only comprises about 5% of the total number of liver transplants that we do in the United States. And there are really only a handful of centers, 10 or so in the United States that are considered high volume centers who do 10 or more living donor liver transplants per year. Next slide. This is a map of the country, and the stars are showing you where the centers are that are considered high volume centers. And University of Colorado not only performed the first adult to adult living donor liver transplant in the United States, and quite frankly, the University of Colorado back in the 60s was the one to do the very first liver transplant in the United States, period. That was Dr. Tom Starzl. but we are considered a high volume center for living donor liver transplantation, and really the only one in our region outside of, you can see there's some centers in Minnesota and Chicago and Texas, as far as the Midwest goes. Next slide. So I've been at the University of Colorado 4 years now in August, and you can see our volumes have grown nicely for liver transplantation over that period of time for both adult and pediatric liver transplantation and for living donor liver transplantation. Next slide. With living donor liver transplantation, unlike a kidney transplant where we really have to match the immunology very closely, with liver transplantation, it's more about the anatomy of the liver of the donor and the size that we need. So for instance, for a baby or a pediatric recipient, we typically only need a small piece of the donor's liver, a portion of the left lobe of the liver, which comprises about 20% of the donor's liver. Next slide. And this is the very first child who, you can see the picture on the left. The mom was the donor to her little girl. And this was done back in 1989 at the University of Chicago by Dr. Chris Brolsch, who I studied with, who's shown there in scrubs. And this is her invitation to her graduation in 2006. And I can tell you she's still doing very well. She's gotten married and she's just had her second child. So the outcomes after living donor liver transplantation are outstanding. Next slide. For an adult, we need a much larger portion of liver tissue, and typically we need about 60%, which is the right lobe of the liver is in most people about 60% of their liver volume. So if it's a typical adult to adult transplant, we're typically taking the right lobe of the donor, which is about 60%, but sometimes in the case of a very petite recipient, we don't need to use the right lobe from the donor. We can use the left lobe, which is about 40%. So this is all part of what we assess when we're trying to determine what the appropriate thing for that particular recipient is. Next slide. Donor safety is our most important and is absolutely our paramount concern. So this is a healthy person who's undergoing a major liver operation with absolutely no benefit to themselves other than, perhaps helping their loved one. So the factors that are important in terms of donor safety is program experience. You certainly want to have your living donor transplant done at a program that has experience, and the cutoff number seems to be that the program has done at least 20 of these. And so there's tremendous differences if the program's done less than that. So about an 80% likelihood that the recipient will not do well if it's an inexperienced program. The selection of the donors, it's a very, very careful selection process. It's an evaluation that's as extensive as what the recipients go through. And then how much liver tissue we need to take from that donor has a tremendous amount of impact in terms of how the recipient does and what their likelihood of complications are, how the donor does, I'm sorry. Next slide.
Hey, Liz, I'm just letting you know we have about 5 minutes.
Okay. There are all different groups of people that see the donors, including an independent donor advocate. We have lots of fancy imaging studies that we can do to estimate the size of the liver. And if we go to the next slide, you can see what these look like. This is a real donor liver. Looking at all of the different blood vessels and bile ducts there. And then the next slide will show you how we can actually do a virtual resection of that liver so that I can estimate how big that graft would be and how much we're leaving the donor with. Next slide. We need to to do an evaluation of the donor that's very extensive, but not every donor needs to have a liver biopsy, although some will require a liver biopsy. And often one of our criteria for specifically for patients with PSC is that if they have a first-degree relative who's donating to them, we would do a liver biopsy prior to donation to make sure that that liver looks healthy. New slide. We need to leave the donor with at least 30% of their original liver volume. New slide. And as I said, the amount of liver tissue that we take from the donor is the single most important factor that predicts outcomes. So the bigger the amount of liver we have to take, the more risk associated with that operation and the longer time it is to recover. Next slide. So this is just a real live picture of what the liver looks like when it's split in the donor, which is down in that lower right side. Next slide. The most important thing I want you to get out of this is that if you have the option of a living donor liver transplant, pursue it, because your likelihood of dying waiting is cut in half and your outcomes are significantly better than patients who get a deceased donor transplant. Next slide. And here's the outcome slide. So you can see the red line is the living donor recipients and their likelihood of surviving out, 2, 4, 5, 10 years is better than receiving a deceased donor organ. Next slide. Now I just want to turn our attention in these last couple of minutes to altruism in Colorado because it's really extraordinary. There are many examples of how people in Colorado are much more altruistic than most of the rest of the country. And here's one. So if you look at who signs up to be an organ donor, Colorado leads the country with almost 70% of people signing up to be an organ donor. Next slide. We have had a tremendous opportunity with people who have contacted us who are interested in donating a portion of their liver to someone that they don't know, but someone who's awaiting a liver transplant and doesn't have a living donor. We started to do this in 2017. At this point, we've had 38 potential donors who had no one in particular that they wanted to donate to. We put them all through the evaluation, and 19 have actually gone on to complete donation for someone they don't know. We have another one scheduled. Amazingly, 11 of these people have also donated a kidney, 10 of them prior to their liver donation and 1 after liver donation. Next slide. Let's go to the next slide. So I just want you to look at our recipients of non-directed anonymous donations. So in 2019, we had 6 pediatric recipients and 3 adult recipients. So far in 2020, we've had 4 pediatric and 2 adult. And as I said, we have another pediatric scheduled for next week. So this is very different than the rest of the country. The incidence of anonymous non-directed donation in the rest of the country is less than 1%, and at the University of Colorado, it's about 12%. Next slide. So you can see as far as deceased donor and living donor transplants over at Children's Hospital, in 2019, we did as many we did more living donor transplants than we did deceased donor transplants, and many of them in the orange there were non-directed, so people who had no relationship to the child whatsoever, but just wanted to donate to somebody on the list. Next slide. And as a result of that, we have been able to completely eliminate the wait list over at Children's Hospital. We only have 1 patient who's awaiting transplant because their insurance company will not give approval for a living donor transplant. This is extraordinary. This does not occur anywhere else in the United States. So the patients who have the highest likelihood of dying waiting on the list are children, especially babies. So this is really an extraordinary accomplishment. Next slide. And I just want to end with showing you one of our anonymous donors who, after donation, ended up meeting his recipient. And Aaron is a gentleman who came to me and said that he wanted to donate a portion of his liver. He didn't care whether it was to a child or an adult. At the time, Manny, who you see there, was awaiting transplant, was getting sicker and sicker, and there was really no option for a living donor in his family. And so Aaron donated to him, and they've become very close friends since then. And Manny continues to do well now, about 3.5 years out from transplant. And that ends my presentation. Thank you.
Thank you, Liz. You could talk about transplant all day, so sorry for such a short amount, but I think everyone got a lot of really great information from that. So we're going to now move gears more toward clinical research. So it's my pleasure to introduce Dr. Veronica Miller. Dr. Miller is the Executive Director of the Forum of Collaborative Research and a professor at UC Berkeley School of Public Health. She is a leading expert in regulatory science and policy with the goal of advancing therapies and diagnostics for unmet medical needs.
Hi, thank you so much, Lisa, for that wonderful introduction. And also I want to really say how much that previous talk has just really spoken to me, as I'm sure it has spoken to everyone, in terms of why people do altruistic things, and what congratulations to all of Colorado for having this wonderful statistic. So I will be talking about what is participating in clinical trials all about. Next slide, please. We will start, here are my disclosures. I will show all of the companies that are involved in sponsoring the Liver Forum, of which the PSC Forum is a part, at the end of the talk. Next slide, please. So I'll start with some basics about clinical research and why we do it. We'll discuss some challenges that are specific to PSC. We'll talk a little bit about why one should consider volunteering for a clinical trial. And then we'll talk about building the big picture as we move forward. Next slide, please. So when we talk about drug development and regulatory science, drug development, it's what we do. And by we, in quotation marks here, I'm really talking about all stakeholders coming together, the pharmaceutical sponsors, the patients participating, the caregivers, the clinical researchers and hospitals that participate, the regulators who approve the protocols, and eventually, we hope, actually approve the drugs. So it's really a joint effort. But we want to develop drugs, and the regulatory science part is how we do it. There have to be specific ways in which we do regulatory decisions based on the benefit and risk, and we'll go much more into detail in that. And of course, we do it to improve the health of individual patients and as well as the public health. Next slide, please. So the stakeholders are many of you on this call today, the patients. In this case, we're talking about PSC patients. We're talking about the sponsors, the regulators, and then of course the clinical community. So for patients, it's really the point of view they bring. What is the need? How dire is the unmet medical need? And how much will they be able to accept? What is the acceptability of the benefit and the risk that they have to face? The sponsors, of course, are really important in terms of innovation and bring pragmatism to the equation. And the regulators set the standards for evidence. And then, of course, the clinical community, once a drug is approved, if we have clinicians, we have professional societies, and we have payers. And they're important because they are the ones that will decide what will they prescribe. And the professional societies are the ones that say what will they recommend. And then, of course, the payers will say what we pay for. I think we need to go forward again. We seem to have gone backwards. We need to go forward one slide, please. Yes, and all of that, of course, is based on these individual stakeholders' perception of benefit and risk. So the pillars of regulatory decision-making, next slide. this slide builds. So yeah, just keep going, please, about 3 times. So we have benefit and risk that has to be weighed one against the other, but then we also have uncertainty. Next slide, please. And it's just go back. Oh, yeah. Oh, okay. There we go. Yeah. So it's a very careful balance that we then have to consider. First of all, how much benefit are we really sure the new therapeutic brings? Do we really know how much risk there is for long-term adverse events? And then the level of uncertainty, what we know and what we don't know, also comes into that equation. And sometimes it's quite a trick to balance all of that. Next slide, please. So as I said, benefit, people are going to say how much benefit is enough benefit, risk, how much risk is too much, and uncertainty, how much uncertainty can we live with? And who makes these decisions? Because everyone that's coming into the setting will have a different perspective in terms of how much benefit is enough, how much risk is too much, and how much uncertainty we can live with. Next slide, please. So I love this paper by Janet Woodcock, who is the director for the Center for Drug Evaluation and Research at the FDA, although she's currently been pulled off into the COVID program. But this paper of hers really, I think, explains it very well. We all want new drugs faster. We want better and newer drugs and safer drugs, and we want them faster. And we often don't understand why it takes so long to get a new drug into the market. But it really is a trade-off between how quickly patients can access a drug and the certainty. In other words, the certainty, of course, is the opposite of uncertainty. So she says in this article, drug development can be described as the progressive reduction in uncertainty about the effects of a molecule in people. And as you know, in terms of molecule, that would be your new drug. Next slide, please. And this is my own homemade graphic to kind of illustrate that. So here we talk about the degree of certainty. So the higher up we are, the more certainty we have about the benefit and the risk. And of course, when we first start early on in a new drug's development path, we will not have that much certainty, but at the end, we will have more certainty. And I don't think we ever have 100% certainty, but certainly a lot more at the end of the development program than we have at the beginning. But if you're the parent of a child with a rapidly progressing rare disease, you might be willing to live with that much uncertainty, or maybe even this much, if there's just a little bit of hope that this drug would work. If you're a manufacturer in research and development or marketing, you may have a totally different perception. If you're a regulator, you may feel much more secure about approving something where there's this much evidence in terms of certainty. And if you're a payer, of course, you will have your own ways of calculating what should be reimbursed and how, depending on how certain we are of the clinical efficacy. Next slide, please. So the mandate we have as a community of drug developers, of patients and caregivers and clinical researchers is to provide convincing evidence, or as convincing as possible under the circumstances. Next slide. And basically what we talk about when we talk about a drug that works is that improves how a patient feels, functions, and survives. That's the basic outcome that we're trying to improve. And of course, we want the benefit to be higher than the risk, because even if a drug can make a patient feel and function a whole lot better, but carries with it the risk of sudden cardiovascular disease, then that risk may actually not allow the patient to survive very long, and so the risk would be greater than the benefit. So it all comes together in terms of the efficacy and surviving because you're treating the disease, but also surviving because the safety profile of the drug is as good as it could be. Next slide, please. So we want convincing evidence to show that we see improvement in how a patient feels, functions, and survives, that the benefit is greater than the risk. And as always, I want you to all really be aware that the perspective of this will be different depending on which stakeholder group you're coming from or which personal experience you're coming from. Next slide, please. So let's go through the clinical research step by step. Next slide, please. So this is from the FDA website, and they have good information for patients in terms of approval of drugs. They go through 5 steps. Step 1 is discovery and development. This is what happens in the laboratory. Step 2 is preclinical research. This is a lot of laboratory and animal testing to answer some very basic questions about safety. And if these signals are good, then we go into the clinical research phase, and I will elaborate on that a little bit more. Then at some point, the FDA will review all the data, and if appropriate, will approve it. And then after marketing, after the drug is approved and on the market, there's continuous safety monitoring and very often additional studies that are done as well. Next slide, please. We will now focus just on that step 3, the clinical research. People like to talk about phase 1 and phase 2A and phase 2B. I think it's easier to really understand it when we think about the function of these studies. So in the early stages, we have first in human studies. So this is where usually these studies are done in healthy people, where first people receive a single dose, and then they might receive increasing doses. And each time, if the safety signals are good, in other words, if there are no major safety worries, we will go on to the next dose and longer dosing. And it really tells us, how does the drug behave in healthy people? If everything is hunky-dory here, then we go into the proof of concept studies. So these are small trials in people with disease, and they're usually a shorter duration. Here we do dose finding. We kind of have an idea of what an optimal dose should be, but here we can really look at that in more detail. And we start looking at efficacy. We want to see, does the drug actually do what it is intended to do? And very often, if we're lucky, we actually have a surrogate or a biochemical endpoint that we can look at. So we don't have to wait for patients to progress to really long end-stage disease, as we often see in diseases like primary sclerosing cholangitis. So these are smaller trials, and there's usually a series of them where we do dose finding, confirming the dose, etc. If everything here is okay, then we go into confirmatory studies, often called phase 3 studies. And here we confirm the results in larger trials and usually for longer duration. Next click, please. And we can, so initially here, we, if there's this first in human studies, we will only see safety signals that are really big, because these are not very many people we're looking at, and they're monitored very, very carefully. But the only safety signals we're going to pick up here are those that are really huge and occur in most of the patients. Next slide, next click, please. Here, when we are in smaller studies, the proof of concept studies, they're larger than the first in human, but still fairly small. We will see additional safety signals. And next click, please. And then, of course, we will have more safety signals once we do the confirmatory studies. And you can just do the next 2 clicks, please. And here we will have, when we do the proof of concept studies, we will have some efficacy data, because we're really looking, is the drug really doing what it is supposed to do? And in PSC, very often, the first thing we look at is alkaline phosphatase, which is a marker that tells us that something is improving in the picture, in the clinical picture of PSC disease. And of course, we will have more efficacy data in confirmatory studies, because these are more patients, and so we're confirming everything, we're following them for a longer period of time, and we're really, next click please, being able to really figure out what's on the risk side and what's on the benefit side by the time we finish these confirmatory studies. Now, next slide, please. But even then, after a drug is approved, we really only know so little about the drug. This is one of my favorite graphs because it is so simple. at the time that the drug is approved, we know this much. The dark green square tells us how much we actually know about the drug. The rest of this rectangle is really the lifetime of a drug, or over its whole life cycle. So when the drug gets approved, we only know those adverse events, those toxicities that appear fairly often and that appear fairly soon. So they appear at the beginning of treatment, and they also appear often enough that we can actually catch them in all of the patients that are participating in trials. But then, of course, the drugs go on the market. Many, many, many more patients are being treated with them. And very often we find out some surprises once the drug actually is on the market. And that's really what that post-approval period really is so important for, to make sure that we catch any additional safety events. Next slide, please. So this is all very good, and let's go and develop some drugs for PSC. Well, it's quite challenging, actually, and many of you, some of you have already enrolled in a clinical trial, and many of you really know the clinical picture so much better than I could ever describe it. But there's the challenges in terms of drug development are that PSC is a rare disease. Now, that means that there's not that many patients that are available to enroll at any one time or in any one center. Very different from doing a cardiovascular study or another diabetes study where people can easily enroll tens of thousands of patients. And it's a very complicated disease, and we don't really have biochemical markers or laboratory markers that can function as an endpoint that really tells us if this endpoint changes, we know for sure that the clinical picture will change. That takes a lot of work to get to that kind of conclusion. We also were very good at diagnosing later stage disease, but we lack all of the markers for early disease identifiers. And of course, there's a high disease variability and fluctuating disease activity that we already saw in the previous talk. This sort of going up and down, and then sometimes there's a very rapid decline. Next slide, please. So it's really challenging to design a clinical study when you have all of those challenges. And there's a review by Vesterhuis and Hemming-Carlson in the Journal of Gastroenterology 2020 that is a really good overview of clinical trials in PSC. And I'm just quoting from them. They say a majority of the clinical trials conducted in PSC to date have been ill-powered. Now, what does it mean to be ill-powered? That means that the study was not actually designed for many reasons, or sometimes the recruitment failed, to really be able to come with a conclusive conclusion. So if you have too few patients in a trial, you may never get the answer you're actually looking for because you don't have enough statistical power. Next slide, please. And that is really, really a very serious situation, and one that I feel personally very, very strongly about, because we have an ethical framework for clinical research. And there's 7 components of that: value, scientific validity, fair subject selection, favorable benefit risk, independent review, informed consent, and respect. And Emmanuel Wendler and Grady, who wrote this paper in JAMA in the year 2000, say that this, and they're all really top-notch ethicists, that if all of these apply, then that is necessary, insufficient to make that clinical research ethical. But you need all of them, not just 3 or 4 of them. And when we talk about scientific validity and the value of the research, if we have a study that is underfunded, or underpowered, it really leaves us with a lot of patient dedication to clinical trials and no answer. Next slide, please. So I'm not going to give you an overview. I was preparing tables with lots of clinical trials, but there's an excellent review that I would refer you to that really covers all of that. And I thought it would be more interesting to talk about some of the lessons that we have learned thus far. Next slide, please. So First of all, though, let's talk a little bit about what it means to volunteer for a clinical trial. It's a very personal decision, and it really is a discussion that has to take place between the patient, their clinical caregiver, their at-home personal caregiver, their family. It is really something that is very, very personal, and no one should ever feel compelled by either a clinician or family member or friend to be forced to take place in clinical research. This is something that I think really is a fundamental. Next slide, please. So what are some of the benefits, though, if you do decide? So some tangible benefits are that you may have access to an experimental drugs that are actually quite good. And we will talk about some studies that are showing some good results. And this could be through randomization, that you get randomized to the new drug, or it could be that after the study finishes in 12 weeks or 24 weeks, that everyone receives that new drug open label. So it might be a way to actually get some of these experimental drugs that have a lot of promise sooner. And of course, when you're part of a clinical trial, you're very closely monitored. And many people see that as a benefit. There are also intangible benefits, and one of them is contributing to the greater good. And we've seen that in Colorado with the willingness to donate livers and kidneys. But many people feel that this really is very important to them. And that in itself brings a benefit. It's not as tangible as saying you're going to live this many years longer. But for many people, that really brings a lot of satisfaction, and there's a lot of sort of social benefits in that, personal benefits in terms of participating. Many people do it because they want to be a benefit to future patients. And maybe they're thinking that if things happen quickly enough, and if maybe they participate in a clinical trial and are able to get a little bit better, then maybe they could also live longer and then potentially live long enough to get the next best drug and then the following best drugs. So they see themselves as a future patient as well. And many people, and we've seen that in many disease areas, really feel a sense of satisfaction in terms of belonging to a research community, that they're part of this major effort going forward. Next slide, please.
All right, Dr. Miller, just 3 more minutes.
Okay, great. Of course, we have risks. There's clinical potential side effects, and of course, all of the logistics. Next slide, please. Patients are the most precious resource to any clinical drug development program, and patients have risks, have rights, and I've listed them here. Next slide, please. And sometimes the road might seem very rocky and difficult to navigate, but next slide, next click, please. But if we kind of get together and sit down and look at what we have learned, maybe that can be of benefit to all of us. Next slide, please. The more we know. Next slide. So this is just a diagram from that review I mentioned, just kind of showing what we have learned in terms of UDCA, nor-UDCA, FXR antagonists. And we do see that signal we're looking for in terms of alkaline phosphatase. We've looked at immune modulation therapy, and we haven't seen that signal. So this is very important for us to move forward to kind of understand more about how these drugs work, and build our future research on that. Next slide, please. And we can learn step by step from other diseases. For example, OCA, we have a lot of data in PBC patients and in NASH patients. So we can know a lot about the safety of that drug by looking at what's happening there. But in PSC, for example, we have started with UDCA, but now we have nor-UDCA. And we've started with steroidal FXR agonists, and now we also have non-steroidal FXR agonists. And with each step, we're learning more. Next slide, please. And I will not go through these slides in detail because I'm running out of time, but they will be available for for when you go online, but basically showing the very strong response to Orso or UDCA. Next slide, please. And here we're looking at OCA, which is a steroidal FXR and agonist, antagonist. And then here we have sulofixor, which is a non-steroidal, which seems to be giving us a better signal with a very, very good safety profile. So we're building on the knowledge we have, and we need to keep doing that as a community. Next slide, please. And sometimes there will be a few bumps in the road. And next slide, but we're in it for the long run. So with that, I would like to thank you for your attention. The rest of the slides are acknowledgements. We can just click through them, please. There's the steering committee of the PSC forum, and then all the companies that are supporting us. So you can just click through these, please. And then with that, I'm done, and I thank you.
Thank you, Dr. Meller. That was a really informative talk and really leads nicely to our last speaker of the day, which is Dr. Gideon Hirschfield. He is a clinician scientist at the Toronto Center for Liver Disease at the University of Toronto. And in addition to having a dedicated autoimmune liver disease practice, Dr. Hirschfield has an interest in developing new therapies for patients with autoimmune disease and in particular PSC. So thanks for joining us, and we look forward to your talk.
Thank you very much, and thank you very much to all the organizers for this very impressive evening webinar. So what I've been tasked with is just to give you some update on PSC Partners supported research and a few future trends, just to give you some concepts of where I think things are moving. And I think the most important message is that you as a community have funded for your own disease over $4 million worth of research and counting. And that's been recognized by patients, by investigators, and has become a very important source of really pump priming to try and help move the field forward. And no one expects that each individual research project will give the answer. But I think, as you heard both about transplant and around trials generally, what we see in medicine is a slow movement in the right direction as we try and solve problems. Next slide. So my disclosures and the example of the different sponsors that I've worked with. Next slide. So to put it into context, it's very important. This disease that we're talking about tonight, primary sclerosing cholangitis, sits within a family of rare autoimmune liver diseases. The disease is a disease of large bile ducts. It has distinctions because it's not just the bile ducts that are inflamed, but it's quite frequently the colon that's also inflamed. And unlike autoimmune hepatitis and PBC, in addition to the sort of liver failure that we have to tackle, we also have this ominous risk of cancer, which can be of the bile ducts or the colon. And sadly, to date, transplantation is our best and most effective treatment. But the goal is to prevent transplant and to prevent liver cancer and bile duct cancer. Next slide. And in our clinical practice, what we're always focusing the way that we care for our patients is a chain of events to making a good diagnosis, understanding the scientific basis for disease and what happens, and ultimately, therefore, having therapies. And to get the best therapies requires the best science, and to get the best science requires we understand the disease as much as possible. Next slide. And we need to move forward, but also look back. So in the late 1960s, we have the great Dame Sheila Sherlock summarizing biliary diseases for a medical audience, but really demonstrating that some of the concepts that were thought about in the 60s are still valid to date as to the causes of a biliary disease: abnormal bile salts, infections, malignancy, not developing properly in childhood, immunology, drugs, and chemicals. And some of these themes remain all the way through. And as scientists, what we're always doing is looking forwards and looking backwards. Next slide. The disease that we're faced with, however, is very complicated, and it's a potpourri of reasons as to why one person gets disease and why one person does differently to another. And that needs to be unteased to identify the therapies, and also to recognize that probably, whilst we only have one name for primary sclerosing cholangitis, it's probably a mixture of diseases, and therefore there may be different treatments for different people at different times. Next slide. This is the disease inside your bile duct, ulcerative cholangitis, it looks remarkably like the ulcerative colitis that you may also have in your colon. And you can see how sore the bile ducts are and how much we need to soothe the immune system and the scarring that then results. Next slide. And the disease is increasingly better understood. And this is just a summary slide. It's available for free if you go to the Journal of Hepatology. It's a poster on the 6 C's of primary sclerosing cholangitis that we published earlier this year. And it's also been on Twitter. The disease can be covert, cholangitis, cirrhosis, colitis, cancer. And after all, what we really want, and we can thank Ricky for the 6th C, is a cure, okay? And that's what the research funds that you've raised are all working towards. Next slide. So what I've done is choose just snapshots of the research that's been funded, not because any one is better than another, but because I wanted to show you the breadth of work that's being done and the different approaches being taken. So the first one was a clinical trial, combination treatment of urso with all trans-retinoic acid for primary sclerosing cholangitis. It comes from the AL. Next slide. Why did I highlight this? Because it's an intervention. This funding from PSC Partners didn't pay for the whole trial, but contributed to the whole process. Was the trial a success? Well, that's exactly what you just heard about from Veronica. It didn't actually give the answer that the investigators hoped for, but there were some modest signals about how this particular molecule, which is in fact a nuclear receptor signaling pathway, may have anti-inflammatory effects in PSC. But it demonstrated the will of patients to take part in clinical research, and that we do learn more about each study as we go forward. So when I read the study and I think about the next study, I think about, was the study the right duration? Did it have enough patients? Did it use the right dose of treatment? And were them things that they measured in the study the correct things? So it's impactful, even though it's negative to the first aim. Next slide. What about genetics? So PSC Partners has successfully funded and supported a lot of genetic programs in PSC. This was one, and this actual grant has been renewed in different forms, called the Exome in PSC, and this is from the Netherlands. So remember, this is in Germany. These are international investigators who are coming to PSC Partners for research support. Next slide. Has genetics solved PSC? No, I'm afraid it hasn't. Have we learned something from the studies that have been funded? Yes. We've demonstrated that patients around the world can get together, provide their DNA, and we can have international collaborations on thousands of patients with PSC. We can look at their DNA in different ways, and we can start to align their DNA results to what happens to the patients. Is this particular gene that, as I've just used as this as an example, changed what we do for PSC? No. Do I think it happens to be even correct to this day? Probably not. But it did demonstrate the kind of science that we're headed towards, which is the bigger the sample, the better the clinical information, and we'll come on to registries, the better the chance we have to identify genes that may explain why one person gets mainly cholangitis and another person gets bile duct cancer and a third person has disease that's so mild it never progresses. Next slide. What about cancer? I can't not have a talk on cancer in PSC. It's an ominous thing. It's something that makes patients lose sleep. It's something that makes their clinicians lose sleep. PSC Partner has funded a number of projects around cancer diagnosis. This was a funding to Hanover in Germany, and it was looking at bile and urine as a way to early detect cholangiocarcinoma. Next slide. Why is this important? Well, actually, although the data is very early, there were some signs that using this particular science, looking at bile and urine and looking at the proteins in them, might start to allow us to diagnose cholangiocarcinoma earlier and therefore give patients a choice of different treatments, which of course, as you heard from the talk on transplant, can include transplant if it's timed. This is very difficult, but actually this is an example of commercialization opportunity, because if we are to benefit patients with PSC, we have to work with industry. We have to make sure that any developments that we make ultimately benefit everyone with PSC, wherever you are in North America, Europe, the rest of the world. And so this group has been working to try and improve this test to see whether it's something that could actually be eventually something offered to patients, but it takes time. It might take 5 or 10 years, but you've got to start somewhere. Next slide. What about the animal models that we have? Limited, not perfect, could be better, okay? This is work from Heather Francis, bile acid regulation of mast cells in PSC. Next slide. Again, I'm not going to go into the details, but I want you to understand that where some of the money which was fundraised from PSC Partners, from patients, went to was basic science using animal models of bile duct disease, where they can identify and use sophisticated science to say a particular cell that wasn't thought to be relevant to bile duct disease may be relevant. Why is this important? Because maybe there are drugs that exist already that interfere with this particular cell, the mast cell. It's very important for allergy, in fact, which could then be considered for clinical trials. So again, very basic research. Of course, a mouse is not a human, but it does make progress. Next slide. And then, you know, to end, I want to talk about Jessica's work, Jessica Allegretti, which really had a lot of PSC Partners support about the microbiome and whether that could be a treatment for PSC. Because that's a big theme for where science is going. Whether it can become a treatment, I don't know. It's quite complicated. Your microbiome is not very simple. But next slide. We already know that there's a lot of controversy about drugs like vancomycin in PSC. And vancomycin can improve liver tests. And in fact, vancomycin is a very good treatment for ulcerative colitis in some patients. And of course, there's a lot of interest in whether vancomycin could be changing the microbiome and helping the disease PSC. Next slide. What Jessica did with support from PSC Partners was take a very small number of patients with PSC and give them a stool transplant. Why was this relevant? Because first of all, it showed that it was possible. Second of all, it showed that it was safe. And third of all, there were some suggestions, perhaps, that there was some biology of changing the bugs in your bowel. What does this mean is that in time, actually stool transplants will become tablets, they'll become capsules. The company that's been involved in this particular trial of FMT has really made a capsule already for Clostridium difficile. So maybe there'll be opportunities for trials in PSC, and many people have been trying to get funding for these studies to change the bugs in the bowel to see whether it helps the liver. This wasn't the answer, but it was certainly the beginning. Next slide. So in terms of, you know, where is research headed, you know, what we're faced in clinic with is patients and their MRCP showing PSC and coming to clinic and asking us, where do I stand? What's going to happen to me? Next slide. And, you know, the questions that everyone asks us, is this bad? What should I do? What about cancer? Will Urso help me? Next slide. So where the world is moving in parallel to the basic science in the laboratory is to try and get bigger cohort studies. And I just wanted to highlight this one study that we did from the UK, where we've been looking at patients with PSC and inflammatory bowel disease and using big data sets from across all of England to capture thousands of patients with PSC and compare them to hundreds of thousands of patients with IBD. Next slide. And what Palak's done is therefore been able to look at real patients, not just patients in teaching hospitals, and what happens to them over time in terms of real things, real impactful events, mortality, surgery, cancer, okay? And able to demonstrate things that will help us make the trials better. Our younger patients have a disproportionate burden. Our African Caribbean patients had worse disease. Women had milder disease. Everyone, if you're getting lots of imaging, for whatever reason, it seemed to be good to reduce your risk of dying from cancer. Don't know why, but lots of scans seem to help. Next slide. And we're able then to understand what happens to a patient over time. And again, the details are not that important, but why does this help you as a patient? Because it means that when we go and talk to people like Veronica, and when we go and talk to regulators, and when we talk to industry, and we think about how to design that clinical trial, we try and work out which patients would benefit the most, and what patients do we need to have the right event rate to try and show a difference from a new therapy. Because we aren't going to get a new therapy if we can't show that it works. Next slide. And again, it demonstrates unmet need. Everyone with PSC has challenges, But let me be clear, when you look at the data from across all of England, and I'm sure it's the same in America, that burden really is faced by our patients under the age of 50, okay? They are having a disproportionate impact, PSC, on their quality and quantity of life. And that's where we need to be targeting our new therapies in the first pass. Next slide. And again, it's not just the liver, but it's your colon as well. And our data demonstrated again why we're so keen to improve the diagnosis of even colon cancer in our patients with PSC and colitis. Next slide. And that speaks to trying to prognosticate, tell you what's going to happen. And that speaks to registries, PSC Partners, US registries, Canadian registries, UK registries, Dutch registries, Scandinavian registries, and hopefully we'll come together, because we then get models which help us tell you what's going to happen. But more importantly, again, identify which patients should go into clinical trials. Next slide. So, if we are going to make progress, research is key. Honest research is important. Asking difficult questions is important. And really, if we're going to make a difference for PSC, we'll have to change the whole field that we work in and try and get to our patients earlier when we have a greater chance to make a difference. Okay? We can't wait until you need a liver transplant to develop the next drug for PSC. We've got to be much earlier than that. Next slide. So we're using research, much of which is primed by PSC Partner, to try and get new therapies that are effective, safe, and ultimately change the treatment for you, your family, your partners. Next slide. So thank you, and I hope that demonstrates that the dollars that you raise do make a difference, and that's over time we are inching forward to better treatments and hopefully prevention, cure, and longer life. Thank you.
Thank you. This really has been an incredible and enlightening evening, and now we get to the questions that have previously been submitted and are submitted as we speak. So if all the speakers could turn on their video. And I am going to start with Dr. Pomfret. Two questions about live donation. One, are bile ducts transplanted as part of the live donation? And two, is that 55 age limit for a donor hard and fast?
So the first question about bile ducts, the liver contains bile ducts within it. So bile ducts are transplanted into the recipient. What we try to do is not use the recipient's main bile duct to hook up the bile duct with the new liver graft. So often we'll do a different type of reconstruction for that because of the inherent problem with the recipient's bile ducts. As far as the age goes, 55, we do consider donors between 55 and 60, but it's really on a case-by-case basis. And, you know, it's just that after the age of 60, we know your liver doesn't regenerate as well. And between 55 and 60, very often people have other health issues that come up that preclude them from being an ideal donor.
Is there a cutoff age to be a recipient?
We don't have an absolute cutoff age. I would say that over the age of 70, very often recipients have other medical problems that make it difficult for them to undergo a liver transplant, but we don't have an absolute age cutoff, no.
Thank you. This next question is for Dr. Miller. Many people want to know, how can they determine whether a trial's benefits outweigh the risks?
So that is a personal question in many ways, or a different question depending on the circumstances. Obviously, a trial in an earlier phase, like when we're still in the proof of concept, we don't know as much about the new drug yet. Whereas in a later phase trial, we have a lot more information already and have a pretty good signal as to whether the drug shows some efficacy or not. And so you know, whether there might be an adverse event happening, etc. Those are all things that we find out as the research participates, progresses. In terms of a personal benefit or personal risk, you know, it takes a lot of commitment to be part of a clinical trial. It means going to clinic very often. It might interfere with other aspects of family life or social life, etc., in terms of you know, the individual circumstances for an individual. And then, of course, the ultimate regulatory decision as to whether to approve a new drug, that will depend on the overall benefit or the overall risk that we have seen with that drug, and that decision gets made. But the decision whether to participate in a clinical trial, if we knew that the drug benefit was an absolute given, then we wouldn't be doing the trial. So there's always some risk involved in terms of participating in a clinical trial, because what we're testing is, you know, what is the efficacy and what is the safety of the drug. So I'm sorry I can't give you a very direct formula, but it really boils down to personal circumstances and the individual trial that people are considering.
Next question is for Dr. Hirschfield. How does the Scientific Medical Committee make their recommendations on what grants are chosen to receive funding?
Good question. So, I mean, the committee has a variety of people on it. It has both scientists, patients, and clinicians. And what we do is, you know, the PSC Partners office makes it very clear how to apply for this grant, gives the investigators time to apply. And those grants are all read by a number of people, and they're all scored, and they're scored in the same way as any other academic grant submission. The panel then meets, and the panel looks at the scores and essentially discusses all the grants, spends most of the time on the grants that score highly, and then makes recommendations about whether it's fundable. But actually PSC Partners makes the final decision, because that's very important, because this is PSC Partners money. And you know, the different priorities for a charity are not just necessarily a mouse study, but it could be equally a study which is around education, around the patient video, around quality of life. And so then I think, you know, the executive of PSC Partners then have the more difficult decision to say, well, all these are the ones that we think are fundable, and how should we use our money most judiciously to meet our goals, and to make sure that we are capturing as an organization a variety of projects that reflect the variety of problems that our patients face. Because, you know, if you're a patient and you've got a rare complication of a rare disease, that doesn't mean it's not important to you. And if you're a charity that supports everyone, there'll be times when you say, well, okay, maybe this is only a very small point, but actually it's important enough that we'll fund it because no one else will. So I think that's really testament to the fact that the PSC Partners has gone to a lot of trouble to make the process as objective as possible, but also as open to make sure patients get the greatest benefit.
Okay, back to, I'm gonna have two questions again for you, Dr. Pomfret. The first one is, why can't patients schedule a living donor liver transplant when their PSC symptoms are severe?
Well, as I was explaining in the presentation, we use the MELD score, and as a person gets sicker and sicker, and you know, their disease becomes more and more severe, they may in fact develop other problems besides liver problems. So they may have their kidneys start to fail. We know that a part of a liver doesn't function the same as a whole liver. So sometimes when people are just too sick, we need the horsepower of a whole liver and not just a partial liver.
Why am I told by my doctor that it's too early to talk about liver transplant? Why can't I just get a live donor transplant now, because I know I'm just going to get sicker?
Okay. And again, this comes down to that whole issue of timing of transplant. We don't want to do the transplant prematurely when the person actually has a better survival, better outcome, just not undergoing transplant. than undergoing the transplant, because the transplant has about a 10% chance of the person dying in that first year as a result of complications of the transplant. So we need to look at the total patient. What sort of symptoms have they had? So I'm not saying that you can't have a low MELD score and go ahead with a transplant, because we often have patients with PSC that have MELD scores in the teens, so certainly not high enough to get a deceased donor organ, but they've had recurrent episodes where they've had to be hospitalized for infection or for some other complication related to their PSC. So it's really a very individual discussion based on that patient's labs and the complications that they've had from their liver disease.
Thank you. Dr. Miller, how is my privacy protected if I'm in a trial?
That's a very important question, and indeed. So all of the usual regulations regarding personal health information and personal identifiers are in place. Usually in terms of databases, etc., the patient name is not really used. There will be a unique identifier allocated to each participant. And that is the only thing that is communicated to the sponsor, into the database, etc. So, so in addition to all the normal rules about privacy that pertain to normal clinical care, there are all of these additional efforts that are made to really protect the privacy of the patient.
You know, a lot of patients ask that. They're afraid somehow that they are, their personal data is going to get revealed. And
right, and everything, right, and patient-informed consent should be very explicit about what data will be generated, what data will be used, and what that data will be used for. So all of that should be very well outlined and explained in the patient-informed consent.
Dr. Hirschfield, do you think NASH, or fatty liver disease, has been taking away some funding and interest in studying PSC?
No. You know, at the end of the day, you're right. New drugs cost a lot of money to develop, and companies will have to make real decisions. But actually, PSC patients are benefiting from the investment in inflammatory bowel disease, and they're benefiting from the investment in NASH, because science doesn't just stop at one disease. And the things that we learn from colitis and the things that we learn from NASH actually help develop drugs for PSC. You aren't going to change the fact that PSC is a rare disease. That isn't going to change. You aren't going to be in charge of the global economy, and therefore, you know, it's not worth worrying about that. But I think companies are committed to common diseases and rare diseases. The model can work for both, and PSC has actually been a beneficiary of the investment in NASH and the investment for the equivalent in ulcerative colitis. There may be drugs that are being developed for ulcerative colitis where people have spent tens of billions of dollars that may one day be very good for patients with PSC.
Dr. Pomfret, could you just explain if there's any advantages to getting a live donor from a blood relative versus someone who is not related? Does the immunosuppression change? Is rejection less?
There's no real advantage to getting a liver from somebody that you're genetically related to. And sometimes it's not a good thing, because sometimes, you know, patients who are genetically related can sometimes share disease processes that we often end up diagnosing in the potential donor when we're evaluating them for donation. But we know from different studies that have been done, especially in the pediatric population, that there's no advantage, for instance, for a mom or a dad donating to their child, even though, you know, for genetically related people to donate to one another. You may need less immunosuppression, but you need immunosuppression. The only time you wouldn't need immunosuppression is if your donor was your identical twin, and that's very unusual. So in every other circumstance, we need immunosuppression, and whether, you know, you might need more or less, depending on you're related to the person, we really wouldn't know that. So there's no real advantage.
Dr. Miller, what happens if your doctor doesn't mention clinical trials to you? How can I get involved?
Well, then, first of all, ask your doctor. And if your doctor really doesn't know anything about clinical trials, then go to the PSC Partners website and find out what is happening in terms of clinical research. Ask your doctor to go on the PSC Partners clinical trial portion of the website and really educate themselves about this, because it really is a patient right to participate in clinical research if they choose to do so. And I know in some smaller clinics and hospitals, maybe clinical research is not as active as in Dr. Pomfret's hospital or in Dr. Gideon Hirschfield's hospital setting. But every patient should have the right to participate in clinical research. You can also, of course, go to clinicaltrials.gov, but that, you know, it's reasonably easily navigatable, but, you know, there's an awful lot of information there. So really going to organizations like PSC Partners, I think is a really good idea.
Dr. Hirschfield, in your experience, in your opinion, what drug in the pipeline do you think has the most promise?
That's a very difficult question. I don't think I can answer it. I think what has the greatest promise is A, the fact that industry is prepared to do clinical trials in PSC, B, the fact that patients take part, and C, the fact that there are many drugs in development, because I think the solution to PSC won't be a single drug. I think we'll have to tackle inflammation, we'll have to tackle scarring, we'll have to tackle changes in the bowel bacteria, we'll have to look at bile acid signaling, and we're going to have to put together many pieces of a puzzle to really make a difference, because this is a very complex disease buried away in your liver. Think of it a bit like Crohn's disease of your bile ducts, but you just can't access it. So, you know, I'm just thrilled that there are 2 phase 3 studies in the world, frankly, because they're so hard to do. And I think that, you know, it will be a bit like an accelerator. As we get a bit of positivity, then the drugs will come together, and then the industry will see that they can take the risk and the patients will ultimately benefit. But you've got to hang in there. It's not going to be easy.
And this is a question really for all of you. If you could just comment on how COVID has impacted research. Was any study abandoned? Is research back up again? And has it affected liver transplant in terms of number of organs? So do you want to start with that, Dr. Pomfret, the surgery aspect?
So during, you know, the very beginning of COVID, when we had no idea what to expect, most programs halted certainly their living donor portion of their program. Most of the time, the overall liver transplant program was kept active because of, you know, there being no option, like for instance, dialysis for kidney transplantation. So more kidney programs were suspended during that period of time because we use higher anti-rejection doses in the kidney patients. So our program, you know, had our liver transplant program open throughout, and we actually did living donor liver transplants during the COVID period as well, but not all groups did. Every single donor, whether they're a living donor or a deceased donor, is tested for COVID. And if they are positive for COVID, then we do not use those organs. So it has decreased the total number of potential donors because there have been a number, as everyone knows, of people who have died with COVID infections, whether that was their primary cause of death or they just happened to also be infected with COVID. But those are organs that we are not using at this point.
And what about research? I know that at our center, all of our research trials were put on hold you could not enroll any new patients, but they're up and running now.
That'd be the same for us. I mean, we managed to keep going with studies which were industry trials, which were actually patients recruited. We had a hiatus. It is difficult. We are reopening. We are re-equilibrating. Our patients are being managed virtually, so we have to encourage patients to come, and take part. We're trying to get studies to be a more mixed hybrid of virtual and in-person visits. And there will be an impact, but I think everyone's will is to try and make that impact as small as possible. And we can only hope that with further COVID waves, people are braver to keep things going, because it's important to have non-COVID research as well as to focus on COVID. Otherwise, we may be missing the bigger picture of society.
Right. Yeah, I agree with that. And then some of the other disease areas that we work in, where we have had webinars specifically to find out how the COVID pandemic has affected clinical research, including PSC. And it's really interesting what happens when people are in lockdown. And of course, Dr. Hirschfield, you're lucky you're up in Canada, where things have sort of slowed down and been kept in check. And of course, every place in the world is a little bit different. But when people stay home, the kids don't go to school, life changes. And maybe they would normally get a runny nose, or they would get a cold that they might not get. Or they might eat junk food in the school cafeteria, and at home they eat better. Or vice versa, that at home they just sort of eat comfort food. But it's really the impact on the overall health of people that some clinical centers and some diseases that are really affected by diet and physical activity have noted major overall changes in the health of the trial population, which then when you're looking for something a little bit less tangible than a biomarker, is sometimes then difficult to tease out what's actually the pandemic effect versus the trial effect. But I totally agree, this move, and in fact, I had hoped to be able to talk about it, but I wanted to say so much and I had to cut a bunch of it out. But this idea of moving towards decentralized clinical trials and managing patients virtually, I think, again, we need to make the most of what we can learn from the pandemic and how we're dealing with remote monitoring, and really then be able to make all trials more patient-friendly. And, one of the things patients can really look at when deciding to engage in a clinical trial is the sponsor really taking advantage of things that can be done remotely in a way that's really patient-centric, versus requiring every patient to come to the clinic, even if it's like 80 miles away, every month or whatever the schedules of the visit are. So I think that's another thing where the patient voice really comes in, and patients should demand that we move into more decentralized trials as we learn how to do that through the pandemic. But why go back to the old ways when we can have a hybrid approach and to really benefit everyone?
I think that's kind of a great way to end before I hand this over to Ricky. I think a theme that has run through all three presentations is how it's important to be your own advocate, whether it's in asking about live donor transplant, asking to be enrolled in a clinical trial, and how great PSC Partners has been in funding up their own research to actively participate and voice their concerns about PSC. So thank you all. This has really been a great night. Hopefully everyone has learned, and handing it over to you.
Thank you. Thank you, Drs. Pomfret, Miller, Hirschfield, and Forman, for an absolutely information-packed session today, and for all your thoughtful responses to our community questions. And I'd like to say that as patients and caregivers, we are happy to hear positive updates about the future of PSC research, additional transplantation options, and clinical trial opportunities. I was sorry we had to cut some of you off. You all had so much to say. It was fantastic. So thank you so much. And before we go, I just want to have a few more reminders. Again, please join if you're a patient or caregiver or clinician or researcher. We would like everybody to join our Voices event, Hear Our Voices, on October 23rd. Please, if you're a patient, please take time to fill out our surveys on PSCD, on the registry, and on COVID-19. Join our patient registry, visit links to our updated COVID-19 information, sign up for our mailing list, watch for the final conference webinar, join the Caregiver Zoom room on October 6th, and then don't forget our PSC Awareness Day on October 29th. Thank you again to our wonderful speakers and our conference sponsors, our conference co-hosts, University of Colorado Health and University of Colorado and Children's Hospital Colorado, and also our corporate sponsors, Mirum, Intercept, Gilead, Alvareo, and Shire. Please stay in touch. Thank you.